Under optimal conditions, a single bacterial cell is a perfect replication machine, dividing into two, then four, in rapid, seamless succession. Microbes have adapted to survive in nearly every environment on Earth. Yet, regardless of their evolutionary pedigree, they remain entirely bound by the strict laws of biophysics and thermodynamics. This diagram maps a microbe's survival envelope. Every organism possesses specific cardinal limits, absolute minimum, optimum, and maximum thresholds for environmental factors like temperature, pH, osmolarity, and radiation. When pushed past these maximum biological thresholds, the organism's internal structures mechanically fail. Microbial control is the deliberate, engineered weaponization of these environmental limits. By applying specific physical forces and concentrated chemical agents, we systematically dismantle the metabolic machinery that keeps pathogens alive. Whether the objective is sterilizing a surgical suite or preserving a global food supply, success relies entirely on identifying a pathogen's biophysical breaking point and decisively exceeding it. The most universally applied physical weapon in this arsenal is extreme heat. Microorganisms cannot regulate their internal temperatures, leaving their metabolic machinery at the mercy of their surroundings. As temperatures surge, the folded structures of cellular proteins unspool and denature. Simultaneously, the lipid bilayers forming the cell membrane lose their rigidity and melt. Evolutionary biology offers a complication. Extremophiles in boiling thermal springs evolved heat-stable proteins and ether-linked lipids to survive temperatures that liquefy standard cells. To overcome this intense biological resistance, we turn to a specific engineering solution: pressurized steam. In clinical and laboratory settings, the definitive tool for physical sterilization is the autoclave. By trapping steam under immense pressure, the autoclave forces temperatures up to 121 degrees Celsius. This threshold surpasses the maximum growth temperature of virtually all known pathogens, ensuring total cellular destruction. Conversely, extreme cold serves a different function. Refrigeration is typically bacteriostatic, pausing metabolic growth by solidifying membranes rather than destroying the cell. Managing these precise thermal thresholds has historically been the technical standard for industrial food safety, ensuring that sealed canning jars remain free of lethal spores. Thermal control succeeds by bypassing chemical defenses, applying pure physical force to dismantle structural architecture. Where extreme heat would destroy materials, we deploy an invisible physical weapon: radiation. This diagram maps the electromagnetic spectrum. Shorter wavelengths carry lethal energy. At exactly 260 nanometers, UV radiation fuses adjacent thymine bases, creating fatal DNA kinks. For deeper penetration, extreme ionizing bands like gamma rays physically shatter the DNA backbone into disconnected fragments. Early technological applications of this high-energy radiation demonstrated its capability to penetrate deep into dense materials, targeting hidden microbial loads. There is one notable biological outlier. The bacterium Deinococcus radiodurans survives massive doses of ionizing radiation by meticulously piecing its shattered genome back together. Against nearly all other life forms, radiation functions as a precision weapon. It targets the genetic blueprint directly without needing to dissolve the physical cell wall. Beyond applied physical forces, control is achieved by manipulating the chemical environment, beginning with the availability of liquid water. By surrounding a cell with massive concentrations of solutes like salt or sugar, we manipulate osmolarity, effectively weaponizing water against the microbe. We see this principle operating in extreme natural environments, like the shoreline of the Dead Sea, where high solute concentrations suppress nearly all microbial life. This cross-section illustrates hypertonic shock. The external salt concentration exerts intense osmotic pressure, stripping water from the cell's interior. As rapid dehydration takes hold, the plasma membrane shrinks and violently tears away from the rigid outer cell wall, resulting in fatal plasmolysis. A second vector of chemical destruction relies on the lethal byproducts of cellular respiration. Toxic oxygen derivatives, such as hydrogen peroxide and superoxide radicals, act as chemical burns at the microscopic level, aggressively oxidizing vital cellular proteins and lipids. Human immune cells, like macrophages, synthesize these exact weapons. They envelop a bacterium, executing the invader with a concentrated burst of reactive oxygen species. Chemical control succeeds by shifting the environmental equilibrium so drastically that the cell is methodically dismantled from the outside in. These biophysical and chemical weapons appear flawless in theory, yet in the reality of infected medical implants and fouled industrial pipelines, clinical disinfection routinely fails. This failure occurs because microbes rarely exist as isolated individuals. Instead, they engineer complex multicellular communities known as biofilms. This diagram reveals the architecture of a biofilm. Its first line of defense is the extracellular polymeric substance matrix, a dense shield of carbohydrates, proteins, and DNA. When antibiotics are applied, they are neutralized or trapped in these outer layers, failing to penetrate the core. But this physical barrier is only half the problem. Deep within the oxygen-starved center of the biofilm hides a biological loophole: persister cells. Persisters are not mutants carrying dedicated antibiotic resistance genes. They are metabolically dormant variants that have powered down their cellular machinery in response to starvation. Because most antibiotics and chemical agents disrupt active biological processes, like cell wall synthesis, these dormant cells present no targets. The chemical attack passes over them harmlessly. Once the course of disinfectants is washed away and the environment stabilizes, the surviving persister cell resumes active division and rapidly repopulates the entire colony. Eradicating a biofilm requires a combination of aggressive physical disruption to shatter the matrix, followed by targeted chemical agents to expose the exposed foes. Microbiology is now focused on dismantling the collective resilience and evolutionary architecture of the microbial world.